Hello, everyone. Welcome to today's live broadcast of using engineered particles and capsules for rapid-to-clinic inhalation applications, presented by Pharmaceutical Technology and sponsored by Capsule Gel and Bend Research. For more than 35 years, Bend Research has worked with clients to create value by advancing new medicines that improve human health and to solve their most difficult scientific and technical problems. The company is a leader in novel formulations, including spray-dried dispersion and hot melt extrusion formulations, as well as a controlled release, inhalation, and biotherapeutics technologies. Bend Research is now part of Capsule Gel's Dosage Form Solutions business unit, headquartered in Morristown, New Jersey, providing access to additional technologies. R&D capabilities and global manufacturing infrastructure for the advancement of client compounds from concept through clinical to commercial manufacture. And we have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them into the Q&A box, which can be found by clicking on the red Q&A widget at the bottom of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the small green icon in the upper right-hand corner of the slide window, or by hovering your mouse over the lower right-hand corner and dragging the window to the desired size. The slides will advance automatically during this event. If you have any technical problems with viewing or hearing this presentation, please click on the question mark help widget in the dock at the bottom of your presentation window. And now I would like to introduce Jennifer Markarian, Manufacturing Editor from Pharmaceutical Technology, and your moderator for today's event.
Thank you. I'd like to welcome everyone to our webcast. Today we are joined by Devin Dubose, Technical Group Lead at Bend Research, and Dominique Cadet, Director of Polymer Science at the Research and Development Division of Capsule Gel. Devin is a Technical Group Lead at Bend Research. His responsibilities include spray drying development and scale up for preclinical and clinical projects, and transferring spray drying processes into a current good manufacturing practice environment. He also has expertise with special spray drying projects for biologics and inhalation. Dominique Cadet is the Director of Polymer Science at the Research and Development Division of Capsule Gel. He joined Capsule Gel in 1974 and has been a director since 1990. As head of the Polymer Science Department, he oversees the scientific development of polymers for capsules, improved knowledge of manufacturing processes for two-piece hard capsules, and optimizing capsule properties and applications. Mr. Cadet also has 19 patents. And now I'd like to turn it over to Devin. Are you ready to begin?
Yes, and thank you for the introduction. So my name is Devin Dubose, and I'll guide us through kind of the introduction and particle engineering aspects of this presentation. And Dominic will talk through the capsule sections. But first, we'd like to go through a few Capsule Gel introduction slides to really set the stage for this dry powder for inhalation offering. So on slide 2 shows the two business units of Capsule Gel. So on the left is the hard capsule business unit. So this is a well-known and established part of Capsule Gel, which includes a wide range of capsule products, including DPI HPMC capsules, as well as a variety of encapsulation equipment. On the right is Capsule Gel's newly formed Dosage Form Solutions, or DFS business unit, which is focused on the delivery of new drug products. And so these are two integrated business units that, through this capsule know-how and particle engineering of DFS, can provide a very unique and comprehensive offering. So slide 3 just shows the Capsule Gel organization, just kind of to set the stage where we're calling in from. So Bend Research in Bend, Oregon on the U.S. West Coast is where I currently reside, and the majority of the particle engineering takes place. France, which is where Dominique is based, and where a lot of the capsule innovation and know-how takes place. And so between the France and Bend site, as well as the many other sites shown here, there's really good communication between them to identify common industry problem statements as well as solutions. So slide 4 shows the breadth of technologies that were discussed during the introduction. So things such as bioavailability enhancement, modified and targeted release, inhalation, and nasal delivery, which we'll talk about today, and other specialized applications like things like biotherapeutics. So these 4 technology pillars are kind of the drug product focused areas, and really have expertise on formulation process development, as well as manufacturing capabilities within these different focus areas. And so now focusing on what we'll actually be discussing in today's webinar, we're hoping we'll provide a background and a case study around a platform approach using spray drying in capsules, focused on rapid progression of inhalation formulations to prove safety and efficacy, as well as show the advantages of having formulation science, spray drying, encapsulation, and specialty capsule expertise through a single organization. And several of the highlights we'll get into that highlight the importance of this for a early clinical approach. And although the focus is kind of on preclinical to phase 2A, these technologies are well established and precedented at the commercial setting. So slide 6 provides kind of the holistic offering, and we'll use this as a guide map throughout the presentation as we move through the different kind of focus areas. So starting with things like materials coupled with formulation and process guidance, the equipment side, so the particle engineering through the spray drying platform and the encapsulation, the capsule and device combination, and the all-important characterization. Really, all of these different components need to be considered in parallel because they can all impact either positively or negatively the aerosol performance or physical and chemical stability. So really, kind of the deliverable here is an active or product concept from a client, so a dose, a PK profile, and through taking that active through this flow chart, the output is really a robust formulation and process for clinical trials. So to start with, we'll focus on kind of the product concept and the materials excipient selection and formulation part of this map. So a couple of different physical models of engineered particles that BIND offers. A lot of people look at the spray drying process as ways of producing amorphous particles, and that's obviously true. But through tailored formulation and process options, we can make both crystalline formulations as well as the amorphous formulations depending on the compound properties. So in the upper left-hand corner, for example, you can see inhalable crystalline spray-dried particles, and on the lower right-hand side, you can see just some SEM images of what an amorphous particle might look like. And so that sets the stage for kind of the formulation approach based on compound properties, but also needs to be coupled with impact to the PK target or profile, which is shown on the right-hand side here. So it's basically a modified BCS system overlaid here with solubility versus permeability. So things in the upper right-hand corner like albuterol sulfate, which has high solubility and permeability, the physical state or physical form, amorphous or crystalline, with the high solubility are all expected to be fast, such that you might, both amorphous and crystalline approaches could be used there and not impact the PK profile. In the lower right-hand corner are more BCS class 2 type compound like fluticasone, where you have low solubility but high permeability. You might expect different PK profiles based on an amorphous or crystalline delivery approach or formulation approach. In the upper left-hand corner, you have BCS class 3 like compounds, so high solubility but low permeability. So things like biotherapeutics, where again, both crystalline and amorphous approaches would be expected to have similar PK profiles. And as our model case study today, we chose albuterol based on the API properties and low impact expected for PK for kind of highlighting the platform approach. So when looking at pulmonary excipient selection, The ideal scenario is nothing, as there's a very few, or there's only a handful of excipients that have clinical and toxicology precedence, and even fewer that have commercial precedence. So, safety is paramount in the selection of these formulations, and the approach should always be to keep it simple. But there are reasons why excipients might be added, which are a few of which are highlighted here. So a lot of these compounds are designed to be, very potent and have low delivery doses to the lung. So one reason could be just as a bulking agent for dilution of the dose and filling mechanisms. In another case, it might be for API stabilization, so especially true with biotherapeutics, which need some sort of hydrogen bonding replacer to deal with water as it's removed during the evaporation process from spray drying. Physical stability, so a high glass transition temperature matrix to limit mobility of the drug substance. And another is for particle dispersibility or aerosolization, so a hydrophobic component at the surface of the particle. So a couple examples of bulking or stabilizing sugars are shown there, with lactose as a bulking agent and trehalose as a stabilizing agent for biotherapeutics. And of course, from the surface modification, leucine, hydrophobic amino acid, which is well known in literature, one of which is provided below to improve the dispersibility and aerosolization of dry powder formulations. So moving on to the equipment side of this roadmap, so primarily the spray drying and encapsulation. Slide 11 provides a high-level overview of the spray drying process and the various, I'll call them subunit operations that make up this the process. And we'll just highlight a couple of the parts of the process that are maybe more important of considerations for dry powder formulations for inhalation. So the atomization process. So this is ultimately what's used to define your target particle size. So through the active loading, the excipient loading and selection, coupled with defining the target droplet size, ultimately is what dries down to your kind of final geometric particle size. And so obviously these have to generally be somewhat small to be respirable. The evaporation or drying conditions, so how quickly and under what kind of steady state conditions these particles dry, can define things like the product morphology, water content, and physical states. This is really kind of the thermal mass and energy balances of the spray drying unit. And equally important is the collection of these particles. So trying to aerodynamically separate particles that have been designed to bypass some of the most efficient aerodynamic separation units in being the lungs can be somewhat difficult. So a lot of engineering and design need to be considered with the cyclone, especially upon scale up. And so some of the equipment we'll show in our case study include a couple of different spray drying scales. So on our left-hand side is what we call our lab spray dryer BLD35, which is a custom unit designed and built by Bend Research. And to the right of that is a pilot scale spray dryer, PSD1, or mobile minor scale spray dryer from Nairo. And so some of the things we'll highlight is the bulk sparing and scalable nature of this process. And this, of course, needs to be coupled with the encapsulation unit. So we'll show here is using the Excipient 600. Again, highlights are a bulk sparing process, filling RSDs of less than 3%, which is often the target for filled capsules for inhalation, and the 100% weight check, which makes it an ideal unit for these early clinical applications. Additionally, these units can, especially when open powder handling is required for filling or sampling, can be enclosed in dry boxes that prevent water and microbial contamination, both of which are usually specifications for these, or have specifications for these types of products. And so at this point, I'll hand it over to Dominic to discuss capsules. And although we won't be going into really specific or device discussions, of course, the capsule device combination is extremely important. We'll be showing during our case study a off-the-shelf, commonly known device.
Thank you, Devin. So it's clear that, as you explained, the capsule is one element of the system, and it is important to understand that there are various options when you come to select your capsule. Capsule Gel, in fact, offers 3 different capsules in terms of polymer composition: gelatin, pure HPMC, and HPMC rescheduling agent. A capsule which is dedicated for inhalation is manufactured with a low level of lubricant. When you talk about lubricant, this is what is also called demolding agent, and it's clear that it has an impact on the powder retention. So when we manufacture a capsule for inhalation, they do have low lubricant level. Of course, these capsules are optimized, as I will show in the next slides, for an optimal puncturing. I will explain what's important there. And there are 2 key characteristics on top of that. One, of course, you already mentioned, Devon, is the weight variation, the weight variability. And the other one, which is more linked to the nature of the polymer, is the moisture content. And of course, for all who know the 2-piece hard capsule, you know that there's a large choice of color, and of course they are printable. Okay, if we look at the characteristic of hard capsule for inhalation in the pharmacopoeia, in fact there is one criteria which we found in the European pharmacopoeia, which is the EP514, where we have specific microbiological requirements on top of the requirements for hard capsule. And of course, this is something which will be checked and which will be reported on the certificate of analysis going together with the capsule. The second point which we add to the certificate of analysis, as I said earlier, is that they are manufactured with a low level of demolding agent, and that's also a mention on the certificate of analysis. When we talk about hard capsule for inhalation, there are also other points to be considered. The size, of course, most of the DPI products come in size 3, a few in size 2s, and I know that tests have been performed in size 4 and 5, but the most common in size 3. Having said that, when you know how difficult it is to optimize the design of the chamber of the inhalation device in order to get appropriate tumbling of the capsule when people inhale, it is important that the dimensional control of the capsule should not be deformed or too thick or too thin. This is important. And also weight, moisture control, puncturing, powder retention are elements which are taken into consideration. So as a consequence of as I said, the importance of this product leads to definition of a kind of customized capsule for each specific product. So just to illustrate a little bit the importance of the choice of capsule in relation with the moisture content, it's clear that a lot of formulation for inhalation may be hygroscopic, may be sensitive to the moisture content, and it is important to know the difference between the gelatin capsule and the HPMC capsule. On the slide here, you have a graph which gives you the moisture equilibrium between the relative humidity of the air and the moisture content of the capsule, expressed as Lawson drying oven method. And as you can see, a gelatin capsule equilibrated at 50% relative humidity, which is a standard, contains approximately 14% water. In comparison, an HPMC capsule contains less than 6 or less than 6%. So already you can see quite a difference. Both capsules being equilibrated with a water activity of 0.5. In both cases, there is a possibility to have capsules with a lower water content, which would have a lower water activity, and we could target something around 0.3, which would give you HPMC capsule where the moisture can be as low as 3% and below. Obviously, if the capsule contains low amount of water, the risk of moisture transfer to the fill is considerably reduced. In relation with the moisture content, you have to keep in mind that this water bound to the polymer may have an impact on the mechanical properties of the polymer. And we'll see that on this slide, you have photos of capsule after puncturing. So the white capsules are gelatin capsules, and the blue capsules are HPMC capsules. On the right-hand side, the capsules are what we call standard. It is the moisture are equilibrated to 50% relative humidity. And you can see that the hole obtained after puncture is very nice, and there is no particle detached. If you look at the picture on the left-hand side, these are capsules which have been stored under dry condition on purpose, and you can see that if there is no significant change visible on the blue HPMC capsule, the opening is nice and the particle is not detached. The gelatin capsule is cracked and you have high risk to have particle. So this will be one element of choice between the HPMC and the gelatin capsule, depending on the nature and the sensitivity of your formulation to moisture. So to conclude the introduction description of the capsule for dry powder inhalation is a specialized capsule to meet specification, and there are different points to be known and to select in order to select the most appropriate capsule. Of course, the nature of the polymer used, gelatin, HPMC or HPMC with gelling agent. We have something to say about weight variability. Even there is an option to have weight check capsule with minimal weight variability. We could talk about custom lubricant level, custom moisture content, and of course, as every capsule, that can be of any color and with any type of imprinting. Kevin, I think it's up to you to continue.
All right, thank you, Dominic. So now we'll proceed to the last part of this roadmap before jumping into the case study, which is some of the different product characterization tools. So slide 20 highlights kind of the analytical toolkit that's common for a lot of these dry powder formulations and their impact on the product profile. So of course imaging, so really just the morphology and lack of agglomeration of these particles, which can be important for the filling process as well as aerosol performance. The all-important particle size and kind of primary focus. So through laser diffraction, it's the geometric particle size of the spray-dried intermediate. And then the aerosol performance, so usually an impaction test, which we'll be showing next generation impaction results throughout the case study, which really the product profile is very product dependent, whether it's deep lung or maybe broader lung deposition. And so this is really, and for those hopefully that majority are familiar with this test, is really a way to kind of rank order the deposition profiles of different formulations, device and capsule combinations, as well as look for lack of change, with different storage times and conditions. So coupled with both particle size and physical stability is the water analysis. So a lot of these formulations can be very sensitive to water uptake, so understanding the equilibrium of the powder, the capsule and packaging combination through things like dynamic vapor absorption, and also understanding what the impact that might have for the aerosol performance and physical stability. And again, coupling things like modulated differential scanning calorimetry, or understanding the glass transition temperatures, as well as the amorphous or crystalline nature of the formulations is a very important analytical toolkit. And so just an example of how we might use this data, and I won't go into all the detail on this slide as it's rather busy, but basically what we're looking at here is an amorphous trehalose and lactose formulation. So what we're looking at is the glass transition temperature on the primary y-axis, and then the water uptake on the secondary axis versus relative humidity. So really just looking at how the glass transition temperature and mobility could change as a function of water content. And this kind of thermal and water analysis really can be used as a guidance for process conditions. So what kind of relative humidity should we target in the spray drying process? Predicted stability. There's a lot of glass transition temperatures and storage temperature models out there that can be used to kind of rank order different excipients. So long-term storage conditions. And really ultimately it's kind of a formulation selection screening tool. And of course, this is a minor part of it and is coupled with, the aerosol performance. So now we'll jump into a case study of how we use all of these different tools in combination to kind of define what our process and formulation should be. So as a model, the model compound we selected was albuterol sulfate, as we mentioned earlier. And so this was formulated as an amorphous dry powder. And this is really the focus of the study. We also will show a one-off later with insulin just to show the applicability for this roadmap for biotherapeutics. The formulation we selected were grass excipients, so lactose as a bulking agent with a high dry glass transition temperature, and trehalose in the case of the insulin to provide hydrogen bonding as it's a non-reducing sugar. Leucine, as we talked about, for aerosol performance and dispersibility, and sodium chloride has the potential to reduce the electrostatic effect of these very high surface area particles. The equipment, we talked about two different spray dryer scales from kind of milligram to kilo type batch sizes. The Excipient 600 for filling. The capsule device combination were Capsule Gel inhalation grade V caps, and the device is a Plastiape RSO1 monodose inhaler, so kind of an off-the-shelf, well-known DPI. And stability, which is ongoing. Of course, everyone wants to see, ensure that the thing remains physically stable and maintains its aerosol performance. So at the moment, we only have 1 month data, but stay tuned for the additional analysis in the future. So for each of the actives we selected for this, we looked at two different active loadings, so a 10 and 40%. And those are kind of chosen to show the applicability of the process to a kind of wide range of active loadings as well as dose ranging options that can be achieved through this platform. So really it's a trait, in the two cases, albuterol sulfate, it's different ratios of active loading to lactose. In the case of insulin, it's different ratio of active loading to trehalose. So in this case, lactose was replaced with trehalose for the large molecules to prevent the Maillard reaction. So slide 24 is some of the initial results that are really highlighting the scalability and bulk sparing nature of the spray drying process, which can be really important as for preclinical studies as well as for future clinical studies. So what we have on the left-hand side are the two spray jars we discussed, and within the table we have two different sprays at the BLD 35 or lab spray drying scale, consisting of a batch size of 300 milligrams on the left and then 5 grams, and then our pilot scale shows a 100 gram batch size. And so the spray drying process, truly is a continuous process. So it's usually dictated by kind of your starting solution volume. So what we see here is a good process yield, which obviously increases with larger batches and encroaches into the 90% range. Similar water contents achievable across scales. And then kind of the aerosol performance results. So the mass median aerodynamic diameter shown there, the standard deviation or span of the distribution, the emitted fraction, and the fine particle fraction, which will be used throughout this this case study for comparing the different formulations. So again, we see very similar aerosol performance in the table there, and then the actual deposition profile from the next generation impaction tests in the graph below. So again, across the different pans or stages of the NGI, we see a very similar deposition profile. And these results are with hand-filled capsules at 10 mg fill weights. So taking it a step further, we're looking at the two different active loadings. So again, the 10% and 40% albuterol active loading with the two spray dryer scales, so the lab scale and the pilot scale. And again, we see similar water contents across the scales. The aerodynamic properties are very similar between active loadings and scales. And again, the overall deposition patterns from the next generation impaction test shown below remain very similar between active loadings and scales. So the takeaway message here is the same aerosol properties can be achieved across a range of active loadings and still maintain its scalability throughout the spray drying process. And again, looking at some of the physical properties and other analytical tests that would be considered for these formulations that we discussed earlier. So the water content, which we've already shown, particle morphology, glass transition temperatures, and X-ray diffraction analysis. And so again, we see similar properties across scales with the 40% albuterol load, and albuterol being at slightly lower TG. We see that decrease in TG, but it's the same across the two different spray drying scales. And some example DSC and X-ray diffraction scans shown below. So slide 27 just shows the SEM images, as everyone likes to look at the actual pictures of the spray dried particles. So again, with the 10 and 40% active loading in the two different scales, we see very similar particle morphology and particle characteristics. And what we do see here is this kind of shriveled raisin morphology, and this is really a function of the leucine used in the formulation, hence why it's added into here. These particles with the more wrinkled morphology kind of limit surface-to-surface interactions between the particles, and like mentioned earlier, are well documented to improve aerosol performance. Similarly, slide 28 shows the geometric particle size across scales, so measured via a Malvern, so laser diffraction unit. And again, we see similar geometric particle sizes between active loadings and scales. So really the combination of all these tests shows just the scalability of different batch sizes, active loadings, formulations, and spray dryer scales. We can keep all the important physical and chemical properties the same. And so moving on to a filling process that's more scalable than hand-filled capsules, we show the Excipient filling results. So here we took the 10% active loading formulations and filled them at 5, 10, and 20 milligram target fill weights. And again, this was done to show the kind of coupled with the different active loadings, the wide dose ranges that can be achievable with this process. So again, in each of the target fill weights, we have an average fill weight very close to the target. We have RSDs less than 3%, and high yields seen across each of the target fill weights. So looks like we have one slide that's missing. Okay, so one slide didn't make it in, but what we did have shown is a comparison of the 10 mg hand-filled capsules, which each of these 5, 10, and 20 mg fill weights from the Excipient, and you have to take my word for it, or we can follow up with this slide deck later. But again, and just like what we showed earlier, is we had the same impaction results, NGI impaction results between the 10 mg and the 5, 10, and 20 mg fill weights on the Excipient. The deposition profiles, again, were very similar. What we do see, and is kind of expected between a 5, 10, and 20 mg fill, is the emitted fraction out of the capsule and device does decrease with higher fill weights, which is kind of expected due to the different surface area to volume ratios. But of course, this plays into some of the mitigation efforts of specialty capsules that Dominic discussed earlier. So slide 31 is kind of the culmination of all of the spray drying and filling results. So what we have here is now the basically the 10 mg fill weights we filled with an exhalidose system across the different spray dryer scales and active loading, so the 10 and 40%. And so again, we're seeing very similar emitted fractions and fine particle fractions, and the deposition profiles in the graph below are very similar. So this is pretty unique in that the same equipment and process conditions can deliver a very wide pulmonary dose range. And so basically what's shown within the different fine particle fractions, emitted fractions, and active loadings and fill weights is a dose range of 300 micrograms to 6 milligrams that can be achieved from a single capsule. So this is pretty important that the same process can get through tox and more with a very wide dosing range, with the key that it does not impact the aerosol performance. And the last slide just shows the incorporation of insulin into this approach. So again, we have our lab scale spray dryer, 2-gram batch sizes, high product recoveries, which can be very important in the early formulation and process development stage, as a lot of these biotherapeutics can be extremely expensive at this stage and beyond. Water contents that are similar across active loadings, and again the aerosol performance, so very similar aerodynamic properties and deposition profiles as shown by the NGI results below. And so again, this is just an additional, throwing an additional molecule into this platform without any additional process development work. So we're pretty confident that this can handle a very wide range of problem statements and drug properties. So the last slide, as part of the case study, shows just how we've kind of set up this study. So right now it's the 10 and 40% albuterol loadings with the focus on physical stability and aerosol performance, as this is a model compound. So these are 10 mg filled capsules of each albuterol loading in glass vials stored in heat-sealed foil pouches, 25 C 60% RH and 40 C 75% RH conditions, and we're planning on carrying this through a year. But at the moment, we only have the 1-month data. So again, we see the different tests that we discussed, so the water content and a little bit of movement seen there, presumably due to different a little equilibration profiles between the powder and capsule. These can easily be mitigated through desiccant selection. Morphology and the glass transition temperature, PXOD results all remain similar, and we're still seeing acceptable aerosol performance. So I guess the takeaway there is stay tuned for future data. So the kind of summary I think that we've tried to show here is kind of through this kind of formulation and process roadmap is really just taking an active and product concept, things like the dose, PK profile, and really defining a product profile through our combined particle engineering and capsule expertise, and of course the delivery of clinical supplies. So kind of how we've looked at these different processes is the feasibility stage, so the formulation identification, proof of concept in early in vivo studies, the process development and scale up in our development facilities and bins, so really defining the process and optimizing for throughput, and supporting dose range finding in GLP toxicology supplies and IND supporting stability. and then finally on to GMP manufacture, which is right next door to our development facility, less than a stone's throw away. And here we highlight the direct transfer of equipment and scale, so that the equipment that we're using in development is the exact same equipment scales we're using for clinical manufacturing, and of course can support GMP stability and release testing. And we have ongoing construction to support commercial expansion in 2015. So thank you everyone for listening and attending today.
Yes, and thank you very much for your interesting presentation. I do want to ask our presenters to stay on to answer some questions from the audience. Our first question is, what are the criteria to choose between the gelatin and HPMC capsules for DPI, and how should one proceed with that?
I think this is for me. The first criteria to consider when you want to decide or to select the most appropriate capsule is really to know if your formulation is hygroscopic and or sensitive to moisture. As I said earlier, the HPMC capsule have a significantly lower water content, and when you receive capsules calibrated to 50% relative humidity, they have 6% water. In comparison, the gelatin capsule have 14%. Both products do have a water activity of 0.5. So that might be a good approach, is to know the water activity of your formulation. And then if it's a low water activity, if it's sensitive to high moisture, there is an option for us to offer you a low water activity capsule, which is valid both for gelatin or for HPMC capsule. We would then deliver a capsule where the water activity would be significantly lower, around 0.3. So I would say to start with, the best thing is to know the sensitivity of the formulation to moisture pickup, and for this water activity measurement would be the appropriate approach.
Okay, thank you. Another question, if you have to change devices somewhere in the human trials, how do the spray-dried engineered particles behave across different DPIs?
Yeah, that's an interesting question. As we kind of looked at the roadmap at the very beginning, we discussed that all of these different components, the formulation, the equipment, and the capsule and DPI combination are critical for the aerosol performance. So that's always a challenge if the need arises to switch inhalers. I think maybe the way I'd answer that is building robustness into the formulation early on, if there's potential for that, might be critical. So maybe that's where I'd focus on the use of something like leucine, which is well known to maybe provide that aerosol dispersibility. So that might be one option to kind of de-risk and provide a more robust formulation that's amenable to a wider range of DPI and capsule combinations in the future.
Okay, good point. Now, what would you say should be the strategy post-phase 2A using engineered particles with capsules?
Yeah, that's a great question. As we said earlier, that the focus for this one was kind of rapid to clinic to prove safety and efficacy. But really, this technology can be applied to well past that. A lot of actually with how highly potent a lot of these drugs are designed to be, in a lot of cases the PSD1 or pilot scale that we discussed in this presentation could be actually your commercial scale spray dryer. For wider or more for certain therapeutic targets, though, that might not be the case, and so the scale up of this to larger scale spray dryers and encapsulation need to be considered. So on the encapsulation side, there's it's in most cases for higher scales, you're switching to a volumetric filling unit, and there's a wide range of companies out there that offer different high throughput encapsulation units that can handle these types of powders that are notoriously hard to handle. On the spray drying side, the spray drying process itself is very well understood by mass and energy balances, but really it's more the equipment side that needs to be considered. So again, the focus on the atomizer, so maintaining the right droplet size that ensures the right particle size, and then the cyclone collection that again has the same collection efficiencies across scales. And there's a few different ways to do that. There's modular approaches that can be used, or of course the right engineering and design can go into the atomizer design as well as the cyclone design.
Okay. Another question: is it possible to deliver high doses via the lung, approximately 100 milligram daily systemic levels, using this technology?
Yeah, I'd say that's one of the big advantages of the spray drying platform, maybe over platforms that require blending and diluting agents for the aerosol performance. So of course the safety profile of any excipients that might be used in the drug are obviously need to be considered. But yeah, so basically we showed a 40% and 10% active loading. There's cases where certain formulations or drug molecules, you might be able to push that as a truly just single spray dried formulation, or loadings as high as 50, 60, 90%. And then that coupled with higher fill weights into the capsule can start to deliver those much higher doses.
Okay, and then when you're talking about hydrophobic compounds in particular, can an API loading of higher than 40% be achieved for hydrophobic compounds?
Yeah, as I mentioned earlier, higher active loadings can definitely be considered within these formulations, balanced again with physical and aerosol performance. In the case here, we've shown kind of aqueous-based spray drying, and obviously there's aqueous and solvent-based systems that can be applied to hydrophobic compounds and maintain higher active loadings, depending on the solubilities of the drug substance and desired excipients.
Okay, and another question we have. The questioner would like to know, is it okay to use pre-filtered atmospheric air as drying air instead of dry nitrogen for DPIs and amorphous solid dispersions?
Well, I guess I'll balance that with my EH&S hat. Yeah, in certain applications, especially with aqueous-based spray drying, compressed HEPA dry air could be used as opposed to nitrogen. That has to be balanced with dust explosion risks and things like that. So of course, it's each process needs to be evaluated for that type of instance. A lot of times for downstream handling, so for like filling operations with the capsules and the packaging, we a lot of times will use just HEPA clean dry air in those cases, as we're not generating large amounts of dust as you might in the spray drying process.
Okay, excellent. Well, it looks like that we don't have any further questions at this point. So we really appreciate you giving your insight on this topic, and I wanted to thank everyone for participating in today's event. I wanted to thank our audience for their questions and their participation, and I definitely want to extend my thanks to our sponsor, Capsule Gel, for making today's educational webcast possible. Please note that this webcast will be available for on-demand viewing through June of 2015, and you will receive an email from Pharmaceutical Technology alerting you when the webcast will be available for replay So please invite your colleagues who may have missed out on the live event to view the on-demand event by forwarding them the email announcement. Once again, thank you for joining us and have a nice day.